5.6 Genetic Code. During replication and transcription, a nucleic acid was copied to form another nucleic acid. Hence, these processes are easy to conceptualize on the basis of complementarity. The process of translation requires transfer of genetic information from a polymer of nucleotides to synthesize a polymer of amino acids. Neither does any complementarity exist between nucleotides and amino acids, nor could any be drawn theoretically. There existed ample evidences, though, to support the notion that change in nucleic acid, that is genetic material, were responsible for change in amino acids in proteins. This led to the proposition of a genetic code that could direct the sequence of amino acids during synthesis of proteins. If determining the biochemical nature of genetic material and the structure of DNA was very exciting, the proposition and deciphering of genetic code were most challenging. In a very true sense, it required involvement of scientists from several disciplines, that is, physicists, organic chemists, biochemists, and geneticists. It was George Gamow, a physicist, who argued that since there are only 4 bases and if they have to code for 20 amino acids, the code should constitute a combination of bases. He suggested that in order to code for all the 20 amino acids, the code should be made up of 3 nucleotides. This was a very bold proposition because a permutation combination of 4 to the power 3, that is 4 by 4 by 4, would generate 64 codons, generating many more codons than required. Providing proof that the codon was a triplet was a more daunting task. The chemical method developed by Hargobind Khurana was instrumental in synthesizing RNA molecules with defined combinations of bases, that is polymers, and that is homopolymers and copolymers. Marshall Nirenberg's cell-free system for protein synthesis finally helped the code to be deciphered. Severo-Ochoa enzyme, that is polynucleotide phosphorylase, was also helpful in polymerizing RNA with defined sequences in a template-independent manner, that is enzymatic synthesis of RNA. Finally, a checkerboard for genetic code was prepared, which is given in table 5.1. The silent features of genetic code are as follows. First, the codon is triplet. 61 codons code for amino acids and 3 codons do not code for any amino acids, hence they function as stop codons. Second, some amino acids are coded by more than one codon, hence the code is degenerate. Third, the codon is read in mRNA in a contiguous fashion. There are no punctuations. Fourth, the code is nearly universal. For example, from bacteria to human, UUU would code for phenylalanine, that is Phe. Some exceptions to this rule have been found in mitochondrial codons and in some protozoans. AUG has dual functions. It codes for methionine, that is mate, and it also acts as initiator codon. Sixth, UAA, UAG, UGA are stop terminator codons. If following is the sequence of nucleotides in mRNA, predict the sequence of amino acid coded by it. 5.6.1 Mutations and Genetic Code. The relationships between genes and DNA are best understood by mutation studies. You have studied about mutation and its effect in chapter 4. Effect of large deletions and rearrangements in a segment of DNA are easy to comprehend. It may result in loss or gain of a gene and so a function. The effect of point mutations will be explained here. A classical example of point mutation is a change of single base pair in the gene for beta globin chain that results in the change of amino acid residue glutamate to valine. It results into a diseased condition called as sickle cell anemia. Effect of point mutations that inserts or deletes a base in structural gene can be better understood by following simple example. Consider a statement that is made up of the following words, each having 3 letters like genetic code. Ram has red cap. If we insert letter B in between has and red and rearrange the statement, it would read as follows. Ram has BRE, DCAP. Similarly, if we insert two letters at the same place, say BI, now it would read RAM has BIR EDC app. Now we insert three letters together, say big. The statement would read RAM has big red cap. The same exercise can be repeated by deleting the letters R, E and D one by one and rearrange the statement to make a triplet word. RAM has EDC app, RAM has DCAP and RAM has cap. The conclusion from the above exercise is very obvious. Insertion or deletion of one or two bases changes the reading frame from the point of insertion or deletion. However, mutations are referred to as frameshift insertion or deletion mutations. Insertion or deletion of three or its multiple bases insert or delete in one or multiple codon, hence one or multiple amino acids, and reading frame remains unaltered from that point onwards. 5.6.2 tRNA, the adapter molecule. From the very beginning of the proposition of code, it was clear to Francis Crick that there has to be a mechanism to read the code and also to link it to the amino acids, because amino acids have no structural specialties to read the code uniquely. He postulated the presence of an adapter molecule that would on one hand read the code and on other hand would bind to specific amino acids. The tRNA, then called sRNA, that is soluble RNA, was known before the genetic code was postulated. However, its role as an adapter molecule was assigned much later. tRNA has an anticodon loop that has bases complementary to the code, and it also has an amino acid acceptor end to which it binds to amino acids. tRNAs are specific for each amino acid as shown in figure 5.12. For initiation, there is another specific tRNA that is referred to as initiator tRNA. There are no tRNAs for loop stop codons. In figure 5.12, the secondary structure of tRNA has been depicted that looks like a clover leaf. In actual structure, the tRNA is a compact molecule which looks like inverted L. 5.7 Translation. Translation refers to the process of polymerization of amino acids to form a polypeptide as shown in figure 5.13. The order and sequence of amino acids are defined by the sequence of bases in the mRNA. The amino acids are joined by a bond which is known as a peptide bond. Formation of a peptide bond requires energy. Therefore, in its first phase itself, amino acids are activated in the presence of ATP and linked to their cognate tRNA, a process commonly called as charging of tRNA or amino acylation of tRNA to be more specific. If two such charged tRNAs are brought close enough, the formation of peptide bond between them would be favored energetically. The presence of a catalyst would enhance the rate of peptide bond formation. The cellular factory responsible for synthesizing proteins is the ribosome. The ribosome consists of structural RNAs and about 80 different proteins. In its inactive state, it exists as two subunits, a large subunit and a small subunit. When the small subunit encounters an mRNA, the process of formation of the mRNA to protein begins. There are two sites in the large subunit for subsequent amino acids to bind to the two and thus be close enough to each other for the formation of a peptide bond. The ribosome also acts as a catalyst, that is 23S rRNA in bacteria is the enzyme ribosome for the formation of peptide bond. A translational unit in mRNA is the sequence of RNA that is flanked by the start codon, that is AUG, and the stop codon and codes for a polypeptide. And mRNA also has some additional sequences that are not translated and are referred as untranslated regions, that is UTR. The UTRs are present at both 5' end before start codon and at 3' end after stop codon. They are required for efficient translation process. For initiation, the ribosome binds to the mRNA at the start codon, that is AUG, that is recognized only by the initiator tRNA. The ribosome proceeds to the elongation phase of protein synthesis. During this stage, complexes composed of an amino acid linked to tRNA sequentially bind to the appropriate codon in mRNA by forming complementary base pairs with the tRNA anticodon. The ribosome moves from codon to codon along the mRNA. Amino acids are added one by one, translated into polypeptide sequences dictated by DNA and represented by mRNA. At the end, a release factor binds to the stop codon, terminating translation and releasing the complete polypeptide from the ribosome. 5.8 Regulation of gene expression. Regulation of gene expression refers to a very broad term that may occur at various levels. Considering that gene expression results in the formation of a polypeptide, it can be regulated at several levels. In eukaryotes, the regulation could be exerted at first, transcriptional level, that is formation of primary transcript. Second, processing level, that is regulation of splicing. Third, transport of mRNA from nucleus to the cytoplasm. And fourth, translational level. The genes in a cell are expressed to perform a particular function or a set of functions. For example, if an enzyme called beta-galactosidase is synthesized by E. coli, it is used to catalyze the hydrolysis of a disaccharide, lactose, into galactose and glucose. The bacteria use them as a source of energy. Hence, if the bacteria do not have lactose around them to be utilized for energy source, they would no longer require the synthesis of enzyme beta-galactosidase. Therefore, in simple terms, it is the metabolic, physiological or environmental conditions that regulate the expression of genes. The development and differentiation of embryo into adult organisms are also as a result of the coordinated regulation of expression of several sets of genes. In prokaryotes, control of the rate of transcriptional initiation is the predominant site for control of gene expression. In a transcription unit, the activity of RNA polymerase at a given promoter is in turn regulated by interaction with accessory proteins, which affect its ability to recognize start sites. These regulatory proteins can act both positively, that is activators, and negatively, that is repressors. The accessibility of promoter regions of prokaryotic DNA is in many cases regulated by the interaction of proteins with sequences termed operators. The operator region is adjacent to the promoter elements in most operons, and in most cases the sequences of the operator bind a repressor protein. Each operon has its specific operator and specific repressor. For example, LAC operator is present only in the LAC operon and it interacts specifically with LAC repressor only. 5.8.1 The LAC operon. The elucidation of the LAC operon was also a result of a close association between a genetist Francois Jacob and a biochemist Jacques Monod. They were the first to elucidate a transcriptionally regulated system. In LAC operon, here LAC refers to lactose. A polycistronic structural gene is regulated by a common promoter and regulatory genes. Such arrangement is very common in bacteria and is referred to as operon. To name few such examples: LAC operon, trip operon, ERA operon, his operon, wall operon, etc. The LAC operon consists of one regulatory gene, that is the I gene. Here the term I does not refer to inducer, rather it is derived from the word inhibitor, and three structural genes Z, Y and A. The I gene codes for the repressor of the LAC operon. The Z gene codes for the beta-galactosidase, which is primarily responsible for the hydrolysis of the disaccharide lactose into its monomeric units, galactose and glucose. The Y gene codes for the permease, which increases permeability of the cell to beta-galactosides. The A gene encodes a trans-acetylase. Hence, all the three gene products in LAC operon are required for metabolism of lactose. In most other operons as well, the genes present in the operon are needed together to function in the same or related metabolic pathway as shown in figure 5.14. Lactose is the substrate for the enzyme beta-galactosidase and it regulates switching on and off of the operon. Hence, it is termed as inducer. In the absence of a preferred carbon source, such as glucose, if lactose is provided in the growth medium of the bacteria, the lactose is transported into the cells through the action of permease. Remember, a very low level of expression of lac operon has to be present in the cell all the time. Otherwise, lactose cannot enter the cells. The lactose then induces the operon in the following manner. The repressor of the operon is synthesized from the I gene. The repressor protein binds to the operator region of the operon and prevents RNA polymerase from transcribing the operon. In the presence of an inducer, such as lactose or allolactose, the repressor is inactivated by interaction with the inducer. This allows RNA polymerase access to the promoter and transcription proceeds as shown in figure 5.14. Essentially, regulation of lac operon can also be visualized as regulation of enzyme synthesis by its substrate. Remember, glucose or galactose cannot act as inducers for lac operon. Can you think for how long the lac operon would be expressed in the presence of lactose? Regulation of lac operon by repressor is referred to as negative regulation. Lac operon is under control of positive regulation as well. But it is beyond the scope of discussion at this level. 5.9 Human Genome Project. In the preceding sections, you have learned that it is the sequence of bases in DNA that determines the genetic information of a given organism. In other words, genetic makeup of an organism or an individual lies in the DNA sequences. If two individuals differ, then their DNA sequences should also be different, at least at some places. These assumptions lead to the quest of finding out the complete DNA sequence of human genome. With the establishment of genetic engineering techniques where it was possible to isolate and clone any piece of DNA and availability of simple and fast techniques for determining DNA sequences, a very ambitious project of sequencing human genome was launched in the year 1990. Human Genome Project, that is HGP, was called a mega project. You can imagine the magnitude and requirements for the project if we simply define the aims of the project as follows. Human genome is said to have approximately 3 into 10 raised to 9 base pair, and if the cost of sequencing required is $3 per base pair, the estimated cost in the beginning, the total estimated cost of the project would be approximately $9 billion. Further, if the obtained sequences were to be stored in the typed form in books, and if each page of book contained 1000 letters and each book contained 1000 pages, then 3300 such books would be required to store the information of DNA sequence from a single human cell. The enormous amount of data expected to be generated also necessitated the use of high-speed computational devices for data storage and retrieval and analysis. HGP was closely associated with the rapid development of a new area in biology called bioinformatics. Goals of HGP. Some of the important goals of HGP were as follows. First, identify all the approximately 20,000 to 25,000 genes in human DNA. Second, determine the sequences of the 3 billion chemical base pairs that make up the human DNA. Third, store this information in databases. Fourth, improve tools for data analysis. Fifth, transfer related technologies to other sectors such as industries. And sixth, address the ethical, legal and social issues, that is ELSI, that may arise from the project. The Human Genome Project was a 13-year project coordinated by the US Department of Energy and the National Institute of Health. During the early years of the HGP, the Wellcome Trust, that is UK, became a major partner. Additional contributions came from Japan, France, Germany, China, and others. The project was completed in 2003. Knowledge about the effects of DNA variations among individuals can lead to revolutionary new ways to diagnose, treat, and someday prevent the thousands of disorders that affect human beings. Besides providing clues to understanding human biology, learning about non-human organisms, DNA sequences can lead to an understanding of their natural capabilities that can be applied toward solving challenges in healthcare, agricultural, in energy production, environmental remediation. Many non-human model organisms such as bacteria, yeast, Caenorhabditis elegans, a free-living non-pathogenic nematode, Drosophila, that is the fruit fly, plants, that is rice and Arabidopsis, etc. have also been sequenced. Methodologies. The methods involved two major approaches. One approach focused on identifying all the genes that are expressed as RNA, referred to as expressed sequence tags, that is ESTs. The other took the blind approach of simply sequencing the whole set of genome that contain all the coding and non-coding sequence, and later assigning different regions in the sequence with functions, that is a term referred to as sequence annotation. For sequencing, the total DNA from a cell is isolated and converted into random fragments of relatively smaller sizes. That is, recall DNA is a very long polymer, and there are technical limitations in sequencing very long pieces of DNA and cloned in suitable host using specialized vectors. The cloning resulted into amplification of each piece of DNA fragment so that it subsequently could be sequenced with ease. The commonly used host were bacteria and yeast, and the vectors were called as BAC, bacterial artificial chromosomes, and YAC, that is yeast artificial chromosome. The fragments were sequenced using automated DNA sequencers that worked on the principle of a method developed by Frederick Sanger. Remember, Sanger is also credited for developing method for determination of amino acid sequences in proteins. These sequences were then arranged based on some overlapping regions present in them. This required generation of overlapping fragments for sequencing. Alignment of these sequences was humanly not possible. Therefore, specialized computer-based programs were developed as shown in figure 5.15. These sequences were subsequently annotated and were assigned to each chromosome. The sequence of chromosome 1 was completed only in May 2006. This was the last of the 24 human chromosomes, 22 autosomes, and X and Y to be sequenced. Another challenging task was assigning the genetic and physical maps on the genome. This was generated using information on polymorphism of restriction, endonuclease recognition sites, and some repetitive DNA sequences known as microsatellites. That is one of the applications of polymorphism in repetitive DNA sequences shall be explained in next section of DNA fingerprinting. 5.9.1 Silent features of human genome. Some of the silent observations drawn from Human Genome Project are as follows. First, the human genome contains 3164.7 million base pair. Second, the average gene consists of 3000 bases, but sizes vary greatly, with the largest known human gene being dystrophin at 2.4 million bases. Third, the total number of genes is estimated at 30,000, much lower than previous estimates of 80,000 to 1,40,000 genes. Almost all, that is 99.9% nucleotide bases are exactly the same in all people. Fourth one, the functions are unknown for over 50% of the discovered genes. Fifth one, less than 2% of the genome codes for proteins. Sixth, repeated sequences make up very large portion of the human genome. Seventh, repetitive sequences are stretches of DNA sequences that are repeated many times. sometimes 100 to 1000 times. They are thought to have no direct coding functions, but they shed light on chromosome structure, dynamics, and evolution. Eighth one, chromosome 1 has most genes, that is 2968, and the Y has the fewest one, that is 231. The last one, scientists have identified about 1.4 million locations where single base DNA differences, that is SNPs, single nucleotide polymorphism, pronounced as SNPs, occur in humans. This information promises to revolutionize the process of finding chromosomal locations for disease-associated sequences and tracing human history. 5.9.2 Applications and Future Challenges. Deriving meaningful knowledge from the DNA sequences will define research through the coming decades, leading to our understanding of biological systems. This enormous task will require the expertise and creativity of tens of thousands of scientists from various disciplines in both the public and private sector worldwide. One of the greatest impacts of having the HG sequence may well be enabling a radically new approach to biological research. In the past, researchers studied one or a few genes at a time. With whole genome sequences and new high throughput technologies, we can approach questions systematically and on a much broader scale. They can study all the genes in a genome, for example, all the transcripts in a particular tissue or organ or tumor, or how tens of thousands of genes and proteins work together in interconnected networks to orchestrate the chemistry of life. 5.10 DNA fingerprinting. As stated in the preceding section, 99.9% of the base sequence among humans is the same. Assuming human genome as 3 into 10 raised to 9 base pair, in how many base sequences would there be differences? It is these differences in sequence of DNA which make every individual unique in their phenotypic appearance. If one aims to find out genetic differences between two individuals or among individuals of a population, sequencing the DNA every time would be a daunting and expensive task. Imagine trying to compare two sets of 3 into 10 raised to 6 base pairs. DNA fingerprinting is a very quick way to compare the DNA sequences of any two individuals. DNA fingerprinting involves identifying differences in some specific regions in DNA sequence called as repetitive DNA. Because in these sequences, a small stretch of DNA is repeated many times. These repetitive DNA are separated from bulk genomic DNA as different peaks during density gradient configuration. The bulk DNA forms a major peak and the other small peaks are referred to as satellite DNA. Depending on base composition, that is A as to T rich or G as to C rich, length of segment and number of repetitive units, the satellite DNA is classified into many categories such as microsatellites, mini satellites, etc. These sequences normally do not code for any proteins, but they form a large portion of human genome. These sequences show high degree of polymorphism and form the basis of DNA fingerprinting. Seize DNA from every tissue such as blood, hair follicle, skin, bone, saliva, sperm, etc. from an individual show the same degree of polymorphism. They become very useful identification tool in forensic applications. Further, as polymorphism are inheritable from parents to children, DNA fingerprinting is the basis of paternity testing in case of disputes. As polymorphism in DNA sequence is the basis of genetic mapping of human genome as well as of DNA fingerprinting, it is essential that we understand what DNA polymorphism means in simple terms. Polymorphism, that is variation at genetic level, arises due to mutations. Recall different kinds of mutations and their effects that you have already studied in chapter 4 and in the preceding sections in this chapter. New mutations may arise in an individual either in somatic cells or in the germ cells, that is cells that generate gametes in sexual reproducing organisms. If germ cell mutation does not seriously impair individuals' ability to have offspring who can transmit the mutation, it can spread to other members of population, that is through sexual reproduction. Allelic. Again recall the definition of alleles from chapter 4. Sequence variation has traditionally been described as a DNA polymorphism if more than one variant, that is allele, at a locus occurs in human population with a frequency greater than 0.01. In simple terms, if an inheritable mutation is observed in a population at higher frequency, it is referred as DNA polymorphism. The probability of such a variation is to be observed in non-coding DNA sequence would be higher as mutations in these sequences may not have any immediate effect or impact in an individual's reproductive ability. These mutations keep on accumulating generation after generation and form one of the bases of variability or polymorphism. There is a variety of different types of polymorphisms ranging from single nucleotide change to very large scale changes. For evolution and speciation, such polymorphism play very important role and you will study these in details at higher classes. The technique of DNA fingerprinting was initially developed by Alec Jeffries. He used a satellite DNA as probe that shows very high degree of polymorphism. It was called as variable number of tandem repeats, that is VNTR. The technique as used earlier involved southern blot hybridization using radio labeled VNTR as a probe. It included first, isolation of DNA, second, digestion of DNA by restriction endonucleases, third, separation of DNA fragments by electrophoresis, fourth, transferring, that is blotting of separated DNA fragments to synthetic membrane such as nitronucleose or nylon, fifth, hybridization using labeled VNTR probe, and sixth, detection of hybridized DNA fragments by autoradiography. A schematic representation of DNA fingerprinting is shown in figure 5.16. The VNTR belongs to a class of satellite DNA referred to as mini-satellite. A small DNA sequence is arranged tandemly in many copy numbers. The copy number varies from chromosome to chromosome in an individual. The numbers of repeat show very high degree of polymorphism. As a result, the size of VNTR varies in size from 0.1 to 20 kb. Consequently, after hybridization with VNTR probe, the autoradiogram gives many bands of differing sizes. These bands give a characteristic pattern for an individual DNA as shown in figure 5.16. It differs from individual to individual in a population except in the case of monozygotic, that is identical twins. The sensitivity of the techniques has been increased by use of polymerase chain reaction, that is PCR. You will study about it in chapter 9. Consequently, DNA from a single cell is enough to perform DNA fingerprinting analysis. In addition to applications in forensic science, it has much wider applications such as in determining population and genetic diversities. Currently, many different probes are used to generate DNA fingerprints. Let us discuss the summary of chapter Molecular Basis of Heritance. Nucleic acid are long polymers of nucleotides. While DNA stores genetic information, RNA mostly helps in transfer and expression of information. Though DNA and RNA both function as genetic material, but DNA being chemically and structurally more stable is a better genetic material. However, RNA is the first to evolve and DNA was derived from RNA. The hallmark of the double-stranded helical structure of DNA is the hydrogen bonding between the bases from opposite strands. The rule is that adenine pairs with thymine through 2H bonds and guanine with cytosine through 3H bonds. This makes one strand complementary to the other. The DNA replicates semi-conservatively. The process is guided by the complementary H bonding. A segment of DNA that codes for RNA may in a simplistic term can be referred as gene. During transcription also, one of the strands of DNA acts as a template to direct the synthesis of complementary RNA. In bacteria, the transcribed mRNA is functional, hence can directly be translated. In eukaryotes, the gene is split. The coding sequences, exons, are interrupted by non-coding sequences, introns. Introns are removed and exons are joined to produce functional RNA by splicing. The messenger RNA contains the base sequences that are read in a combination of 3, that is to make triplet genetic code, to code for an amino acid. The genetic code is read again on the principles of complementarity by tRNA that acts as an adapter molecule. There are specific tRNAs for every amino acid. The tRNA binds to specific amino acid at one end and pairs through H bonding with codes on mRNA through its anticodons. The site of translation, that is protein synthesis, is ribosomes, which bind to mRNA and provide platform for joining of amino acids. One of the rRNA acts as a catalyst for peptide bond formation, which is an example of RNA enzyme, that is ribosome. Translation is a process that has evolved around RNA, indicating that life began around RNA. Since transcription and translation are energetically very expensive processes, these have to be tightly regulated. Regulation of transcription is the primary step for regulation of gene expression. In bacteria, more than one gene is arranged together and regulated in units called as operons. Lac operon is the prototype operon in bacteria, which codes for genes responsible for metabolism of lactose. The operand is regulated by the amount of lactose in the medium where the bacteria are grown. Therefore, this regulation can also be viewed as regulation of enzyme synthesis by its substrate. Human Genome Project was a mega project that aimed to sequence every base in human genome. This project was yielded much new information. Many new areas and avenues have opened up as consequences of project. DNA fingerprinting is a technique to find out variations in individuals of a population at DNA level. It works on the principle of polymorphism in DNA sequences. It has immense applications in the field of forensic science, genetic biodiversity, and evolutionary biology. Thank you for watching this video. Please subscribe and press the bell icon for further videos.